today I'm streaming to you live from my least favorite place in the lab, the cold room. This is like a giant walk-in refrigerated room that's really cold, and I'm in here because I'm doing a Strep Tag Affinity Purification. So basically I have this protein and I had insect cells make it for me. And so earlier this morning, so at about like 5 a.m. I came in and I broke open all those cells, and then I spun them down so that all of the cellular like membranes and gunky stuff gets pelleted out, and then the rest of this was this bright yellow liquid stuff. And so then I combined that bright yellow liquid stuff, so that has a bunch of proteins, including the protein I want. And what I did was I expressed the protein, so when I had made the protein, I had to add on an extra sequence at the end called the Strep Tag. And that binds specifically to this Strep-Tactin resin. The Strep-Tactin resin stuff. So I filled this column with the resin, and I'm flowing the protein through it. Well, actually I combined them first with batch mode, so I combined them in this tubey thing, which I had rolling on the shaker thing. We call this a hot dog roller. It's cool. But anyway, so that I let it all bind, and now I'm flowing it through the column. So there's really only like, I only used 7 milliliters of the resin, but I had like 200 milliliters of my the insect cell lysate. So that's why I did the batch mode first, and now I'm pouring it through. But you can see that the column is, so it's packing, so it's almost flowing through, but it's flowing through pretty slowly. So I can adjust the stop cloud, but it's pretty much basically all the way open. So one of the benefits of using like the ACTA, so like the high pressure chromatography system, is that it can use higher flow rates. But when you use lysates and stuff, it can really gunk up the tubing and cause problems. So I tend to use gravity flow for the affinity step. And so today I want to tell you more about, and then also like, so right now my protein is bound to those little beads, so the resin in the column. And all this other stuff is like the flow through that has all the non-specifically bound proteins. Then what I'm going to do is I'm going to wash it with this wash buffer. And so this is just a buffer. It's the same buffer that I resuspended the cells in. And basically I just want to remove anything that's non-specifically bound to the resin. And then what I'm going to do is I'm actually going to repeat it off with a buffer that's the same as this, but it has a competitor called just Biotin added on. And so that's going to push my protein off of the resin. It's going to like displace it and compete it off. So then I'll have my protein that's mostly pure. And then I'll go forward to further purification steps with ion exchange and gel filtration, assuming all goes well, which I really hope, because we are basically out of media and it's on back order, and yeah, so that's the situation. But now let's talk about the science behind this Strep Tag Affinity Purification and the natural system it's based on, which is biotin and biotin binding proteins, Streptavadin and Avidity. So it's some pretty cool stuff, so let's take a look.
With recombinant protein expression, we take the genetic recipe for a protein we're interested in studying, and we recombine it with a piece of DNA that makes it easier to work with. So often this is a circular piece of DNA called a plasmid for bacteria, or a bacmid for baculovirus for insect cell expression. What happens is that once that plasmid gets, or that bacmid gets into the cells, the genetic recipe gets transcribed, so RNA polymerase makes an mRNA copy of it, and then the ribosomes use those copies to make protein in the process called translation. So if you you can stick in different recipes to get different proteins made, and because you're controlling the sequence you put in, you might as well make it easier for you to purify it out. And a common way we do this is by adding an affinity purification tag, such as the Strep tag or a His tag. This is basically just some extra amino acids, so extra protein letters. So if you put extra DNA letters, then you'll get extra mRNA letters and you'll get extra protein letters, so amino acids. So you have this extra string of amino acids, and then in affinity chromatography, you take this column filled with these little beads called resin. And these resin have are sticky for specifically that tag. So they have like a protein or a metal or something that binds specifically to the tag that you put on. Now you can break open those cells, and when you do that, you'll get a ton of proteins. And so when you break open cells, we call it the lysate. And so then you can flow this lysate through this resin, and your protein will get stuck. Other things will just flow through. Then you can wash all that other gunk through, those other proteins you don't want. And now you have your protein stuck to the resin, so you want to get off the resin so you can use a competitor. So a couple of the main forms of affinity chromatography that I use are a his tag, so that you have a string of the amino acid histidine on the end of your protein, and it'll bind to a nickel-coated column, and you can compete it off with this chemical called imidazole, which looks like histidine, so you can like flood it off. With the strep tag, it has this string of amino acid letters I'll show you in a minute, and it binds to this Strep-Tactin resin, which mimics the protein Streptavadin. And these tags are often designed so that there's a protease recognition sequence. So there's the sequence-specific proteases. So proteases are protein cutters, and if you put the specific sequence that the protease likes to cut in between your tag and your protein, now once you've competed your protein off, you can then cut that tag off and have your tagless protein. You can also do the tag cleavage on the resin, which can be useful in some purposes. So basically, you cleave it while it's still attached to the resin, and then your protein will come off and the tagged part will still be stuck on there. And then you can compete off the tag if you want to reuse the resin. But sometimes that's less efficient that way, so you'll have to like test it out to make sure that it works properly for your protein. So the strep tag is just this extra string of amino acid letters. It happens to be this particular sequence, and it kind of mimics biotin, which is the natural binding partner for Streptavadin. And so Streptavadin is what the Strep-Tactin resin looks like. I actually use this twin strep tag, which has two strep tag sequences in a row with a flexible linker between them. And this is helpful because the Strep-Tactin resin actually has 4 binding sites. And so if you have the twin tag, then that can bind to 2 different sites on the Strep-Tactin resin. So as I said before, Strep-Tactin resin is a mimic of Streptavadin. And Streptavadin is this protein that's made by the certain bacteria to protect itself from other bacteria by binding to the bacteria's to biotin, which the bacteria need to do things to like carboxylate stuff. And so another thing that binds to biotin is the protein Avidin, which you might have heard of. And so Avidin comes from egg whites. And so both Avidin and Streptavadin bind to biotin really, 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 really tightly. We're talking like femtomolar range KD, if that means anything to you. So that's not going to be very useful to us. So Avidin, just in terms of Avidin versus Streptavadin, so Avidin has an extra like part that gets glycosylated, so it has sugar chains added to it. And that can cause it to bind more non-specifically to non-biotin things. So Streptavadin has a little better, selectivity, slightly lower affinity. There's also a form called Nutravadin, which is like a neutral form of Avidin, because Avidin, it's very cationic, so positively charged, and that can cause it to like clump up, which you call like aggregation and stuff. But the thing is, so both Avidin and Streptavadin bind to biotin super tightly. And so this Strep tag actually has, it mimics Streptavadin, but it's in mutated form, so it doesn't bind quite as tightly. And it binds, so it'll bind your protein, but it'll also let you get your protein off of there. And to get it off of there, instead of using biotin, we use this compound called disthiobiotin. So biotin binds to the tag binds less strongly than biotin binds. But biotin binds so strongly, even to this modified form of Strep-Tactin, Streptavadin, that it's really hard to get off. And so it's not very helpful if you have your protein, you compete off your protein, but then you can't regenerate your resin because you can't get that biotin off. So instead we use disthiobiotin, which is a different form, which is it's missing that sulfur part. So thio stands for sulfur, so so without the sulfur part. And it's easier to displace after you put it on the resin. So normally what you do is you flow your protein through. Your protein sticks, everything else flows through. You wash it and wash it and wash it to get anything that's non-specifically stuck on there. And then you elute it by adding disthiobiotin. Normally what I'll do is I'll add some disthiobiotin and I'll let it sit for about 15 minutes, and then to allow it to reach all the resin and everything, and then I'll flow it through. So I typically do that first with like 2 column volumes worth of the Elution buffer, so the buffer containing the disthiobiotin. I'll let that flow through, and then I'll collect that, and that'll have most of my protein in it. And then I'll start collecting like 1 column volume fractions, and seeing how far the protein keeps eluting out. So as I was talking about before, the benefit of having that twin strep tag is that the Strep-Tactin resin has these 4 sites. And this allows for something called avidity. So affinity is the word that we use to describe the strength of an individual binding interaction. Avidity is what happens when you have multiple interactions. So you have multivalent binders. So monovalent has a single valent binding site. Multivalent has multiple binding sites. So since we have that tetramer, we have a tetravalent binding site for the strep tag. And the thing about avidity is it gives you additional strength to an interaction above that you would have with just a single site, or between those sites on their own. So it's like more than if you just had 4 separate sites, because you can think of it like one of them, these sites are constantly being bound and unbound and bound and unbound, and the stronger that they are, the interaction is, the stronger the affinity, the more likely it is either to stick and or to stay stuck, so it's not falling off. If you have multivalent thing, one of the binding sites, if like one of the binding sites is bound, it kind of anchors the other, so even if they come off a little, they'll come right back on. So that helps you, so having that twin tag with that tetravalent site, so you have those 2 sites, and if one gets bound and the other comes flopping up, it'll get flopped back down more easily than if it were to like flow all the way away. So this part gets a little more wonky, but in terms of the affinity I was talking about, so the strep tag and the Streptavadin resin have about a micromolar affinity compared to the Streptavadin and the biotin, which was like femtomolar, so like 10 to the negative 14th, crazy stuff. And so in terms of those numbers they're KD. So the KD is a measure of binding affinity, and I did a whole post on this if people are interested in more. But basically, if you have 2 binding partners, like so you have your tagged protein and you have the beads, And they can bind and unbind and bind and unbind, and you reach this thing called the dynamic equilibrium, where for every one that binds, there's one that unbinds. And we have this number called a KD, which describes the concentration required to reach half maximal binding. So the more that they're attracted to each other, like each chance that the two partners meet, if they're more likely to bind to each other, or if they're less likely to separate, then you're going to have a lower KD. So basically, this means that it's going to take less of the binding partner to reach half maximal, because you're going to hold on tighter to those binding partners you find, or you're going to be more likely to bind them if you run into them. So it can be a combination of the two things. But so when we're talking about high affinity, when you have these lower concentrations required to reach half maximal binding, that's why we have those really, really, really low numbers for the Streptavadin and Biotin. And the Avidin and Biotin is like one of the I think it's the strongest potentially known non-covalent interactions. So two different molecules interacting. As for what it actually looks like, so we take our cells and break them open. So we lyse them. These are insect cells, and in our insect cell systems, it's co-expressing with YFP as a reporter, so that's why it looks yellowy. So we break open the cells and we spin them down to separate the soluble stuff from the insoluble stuff. And so the soluble stuff has a protein, but it also has a bunch of other proteins and other gunk. So we flow it through a column filled with Streptavadin resin. Well, typically what I do is I do the binding in batch mode. So I add it to the resin. I put the resin, add the lysate to the resin, and let it roll on the hot dog roller thing for about half an hour to let it all bind. Because I only have like 5 mil. Usually I have like 5 mils of resin and a couple hundred, 100 to 200 usually milliliters of the insect cell lysate. And so 5 versus 100, I want to make sure that all of the protein gets a chance to bind. And then I flow it through the column and do the washing and get my protein off. And then don't worry, this is how you can regenerate it. So one way to regenerate it is I often use like sodium hydroxide, which will get it off. Another way, and a way to check if your resin is well regenerated, is with this Avidity, and it turns red, but then when you wash it off, your resin will turn white again. And so if the resin turns red if the Avidity is like bound to all the sites, so it's displaced everything. And then when you regenerate it, then it's not red anymore, and it's white and ready to use.